Northeast Missouri State, down Truman State in Missouri. And then she came here for a PhD in physiology. And she worked on reproductive system in chickens. I was reminded of that today with Janice Barr. And then she came over and did a postdoc in our lab on the neural basis of circadian rhythms. And then she took a position in comparative biosciences here and started out on a career of her own, and she sort of established and discovered the field she's going to be talking to you about today. She's now an associate professor and mover and shaker over the pharmacology department at UIS Medical School in Springfield. And she just agreed to come over and tell us what she's been up to. So no one's more excited than me to hear what she's going to tell us. So welcome, Shelley.
So thanks to Martha for inviting me over to talk to your class today. And so what I'd like to start out by saying is that this really is a very new area, I think, in the circadian rhythms field even. A look at the interaction. What we're going to look at today is a lot of interaction between the circadian rhythm, the circadian timing system, and the system that is in place for detoxifying chemicals or things that you might take in that are out in the environment, pesticides, things that you might eat in your diet that aren't easily digestible, things like that. But we're going to look today is how these two systems interact and how this interaction of these two systems, if it doesn't work properly, can lead to disease states. So we'll start here, and I'm going to assume that you know a lot of the basics, and Martha said she gave 6 lectures, so you guys should be up to speed on circadian rhythms. But if there's anything that you don't understand as I'm going through, please just raise your hand or just jump in and ask a question. I'm happy to have a discussion with you about it. So this may be a little bit different clock diagram than what you guys are used to seeing, because I'm sure that most of us, when we talk about rhythms, we put the SCN, the suprachiasmatic nucleus, up there at the top, and everything else is just kind of down here at the bottom. But today, what we're really going to be focused on is things that are really happening and going on in the liver. And so that most of my talk really is about things that are happening in the liver. But more than that, it's about how the liver, the clock in the liver, interacts with the clock in the brain. Right, so most of you should know by now that there is a central clock located in the suprachiasmatic nucleus. It's the master clock. It drives the rhythms in the rest of the body. And then we have a clock down here in the liver. And this clock in the liver, the clock genes are present in the liver. They oscillate with 24-hour periodicity. But typically, the clock in the liver is driven by either the clock in the brain, the SCN, or by food. So there are 2 different ways that the clock in the liver can be regulated, either by the SCN or by feeding. But what I want to focus on is the liver clock itself for just a minute, and the liver itself. The other aspect of what we're going to talk about today is not just clocks, but it's about toxicity. And one of the major functions of the liver is to metabolize chemicals, metabolize drugs that you might be taking, metabolize any sorts of chemicals that are in your body, metabolism of hormones, all sorts of things are broken down in the liver. And so this is a basic diagram that shows how things are broken down in the liver. So we have, and what I'm going to really focus on in my talk today are these things called fat-soluble toxins that might be in the environment. These might be these might be pesticides that are sprayed on farm crops that get into your food. These might be formed from pollution. So in the process of paper bleaching or garbage burning or any other sorts of smokestack that you might see is going to put out chemicals into the environment. And some of those chemicals that are found in the smoke that comes out of there are fat-soluble toxins. And they can get into your body. And once they get into your body, they make their way to the liver. And they're broken down typically in 2 steps. Okay, the first is the production. After these toxins get in the liver, it causes the production of cytochrome P450 enzymes. And these cytochrome P450 enzymes act on the toxins. And what they typically do is they will do one of these 5 things: oxidation, reduction, hydrolysis, hydration, or dehalogenation. So basically, the cytochrome P450 enzymes latch onto these chemicals that come into the body, and they break them down. And what they do is, after they act, these phase 1 P450 enzymes, they work on the toxins and they form an intermediate metabolite. So the chemical comes in, it's a certain structure, the P450 enzyme comes in, adds an oxidative group onto it or causes hydrolysis, and it forms an intermediate metabolite. These intermediate metabolites are sometimes way more toxic than the original compound, right? And so they can be big problems. But typically what happens is the intermediate metabolite is then acted on by another set of enzymes in the liver that will conjugate it. So they put a sulfate group or a glucuronidate group onto this chemical. And basically what this does is it makes it water soluble. And it makes it, so we go from a fat soluble through 2 steps and we end up with a water soluble compound that then is gotten rid of and excreted either in the gallbladder, through the gallbladder, or the kidneys. Okay? So I want to talk a little bit more about one class of these fat-soluble toxins, and they are classified as dioxin. So dioxin is probably the most dangerous, the most toxic chemical known to man. Okay, so what I'm going to show you some structures of dioxins in a couple of slides. But how do you get exposed to dioxin? All right, dioxin exposure can occur by accident. Agent Orange, which was present in Vietnam, was filled with chemicals called dioxins. And there was a big spill in Italy in 1976 where dioxins were just spewed into the environment, so people were exposed to very high levels of dioxins. But more commonly, people and animals are exposed to dioxins in the environment because they can be produced as byproducts of these different types of processes. Incineration, so anytime there's incineration going on to get rid of waste products in the environment, dioxins will be given off as a product of that into the smoke and into the atmosphere. Many pesticides, and in the production of pesticides or the pesticides themselves, and PVCs, production of those. The process of chlorine bleaching can leach out or cause dioxins to be produced. And one of the more common ones is the production of paper. So when they make paper and they bleach paper to be produced, and dioxins are given off as a byproduct of this. And one thing that I don't have up here it's more recent and is in your microwave. So some of the plastic things that you store food in, you put those in the microwave, the plastics will leach dioxins into the food. So there's all sorts of different places where you can be exposed to dioxins. So why is this bad? So we're going to talk a little bit about why this is bad. Well, what happens if you have too much dioxin? Well, this is a list of the things that can go wrong if you get exposed to high levels or dioxins build up in your body. And I should say before I go on is these chemicals, once they're produced, the dioxins, they get into your body and they're not easily broken down by that liver metabolism. The liver metabolism doesn't work very well on them, especially that second phase of liver metabolism. So what you end up with is in your body either the parent dioxin compound or that reactive intermediate, and they build up and they're fat soluble. So they get stored in fat or in places where in the liver or in fat, and so they build up. And it's estimated that the half-life for dioxins in the human body is 10 years. So it's not something that's easy to get rid of. Okay? So I guess my point is, you don't have to have one single high level exposure to dioxin for it to cause a problem. But what more likely occurs in human beings is that you get low levels of dioxin and you're exposed to low levels of dioxin over the course of years, and it builds up and builds up and builds up, and then causes a problem after it reaches a certain level. And these are the kinds of problems that you can get. You can see everything from cancer to neuronal deficits and decreased IQ. There's a professor out at Vet Med who actually studies the effects of PCBs and dioxins on IQ and learning and memory in children. And so she works in that area. But one of the things that I'm going to highlight today in my talk is that dioxin toxicity can lead to diabetes and changes in metabolism and ultimately diabetes, and it can lead to sleep disturbances. And I got really interested in this research area because when I had my faculty position here, there was this old guy that was a toxicologist, and he came up to me one day and he said, "Shelley, in the 70s, I worked with a group of people who were exposed to really high levels of dioxins, and they kept telling me that they couldn't sleep. And well, being a good circadian rhythms biologist that I was, I said, they can't sleep. Well, I wonder if that has something to do with their circadian timing system, because sleep and circadian rhythms are intertwined. So I started to look into that, and that's a lot of what I'm going to tell you today is the result of looking into that process. So, dioxins bind to a receptor called the Aryl hydrocarbon receptor, or the dioxin receptor. All right, so when they come into the body, they're lipophilic, they accumulate in fat, they make it into the liver, they make it lots of different places, and when they get into a cell, they're going to bind to this protein called the Aryl hydrocarbon receptor. And I will tell you right now, as we go on to the next few slides, that this Aryl hydrocarbon receptor is the link between dioxins and the circadian clock. Okay? And I'll tell you why in just a little bit. But just a little bit more about some of these chemicals that will bind to the dioxin receptor. So the dioxin receptor is in the body. It's almost ubiquitously expressed. It's expressed all throughout the body, in the brain, liver, high levels in the gut, in the skin, lots of different places. But it's been classified for a long time as an orphan receptor, meaning nobody really knows what its endogenous ligand is. Okay, but it's been studied for the last 50 years for its binding of these chemicals in the environment and in toxicology. So what kinds of things binds to this receptor? Well, I'll tell you this receptor itself is not very specific. It binds to classes of compounds, not just one specific thing. So you think of the estrogen receptor, for example. It binds really highly to estrogen, and it doesn't really like to bind to anything else. This receptor is not like that. It binds to lots and lots of different things. And it's very ancient. It's really been there for a really long time throughout evolution. And its role is, one of its major roles is when an animal or a person takes in and gets something inside their system that's toxic, they want to get rid of it, right? So the goal of this receptor is to get rid of those things. So, or one of its goals, or one of its functions. So what kinds of things bind to the dioxin receptor? Well, this group of chemicals called halogenated aromatic hydrocarbons. This one TCDD, you should remember that one. There's the structure of it over there. That's the one that's really, 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 really toxic. That's the thing that's in the dioxins that is really, really detrimental to people. But other things like this one, benzoapyrene, is a component of cigarette smoke. So that one will bind to the aryl hydrocarbon receptor. And more recently, what we know is that there are things in your diet, like flavonoids, genistein, which is a flavonoid. It can bind to the aryl hydrocarbon receptor. And of course, for the last 30, 40 years, everybody's been looking for asking the question, what's the endogenous receptor for this? Is there an endogenous receptor for this? That's like the $50 million question in this area. And what we know is, and one of the reasons that we got interested in this was because if you look at this first thing that's known as an endogenous ligand, what you see here is it's a tryptophan photoproduct. Well, if you know anything about circadian rhythms, it's all, we know that light is the major entraining factor for the circadian timing system. So when light hits the body, it does all sorts of things other than just cause regulation of your circadian rhythm. And one of the things that light can do within the skin is the conversion of tryptophan into this compound over here called FICZ. And this FICZ has really, really, really high affinity for the aryl hydrocarbon receptor. So one of the questions that I'm going to get back to is we asked was, does light cause the production of FICZ, which can bind to the aryl hydrocarbon receptor and regulate your circadian rhythm, interact with your circadian rhythm? But I'll get back to that in a minute. For now, I want you to get an appreciation that there's lots of different kinds of compounds that can bind to this particular receptor. All right, so how does it work? So we're focusing still on aryl hydrocarbon receptor and the toxicology now. But the aryl hydrocarbon receptor is a cytoplasmic receptor. When it's not bound to any ligand, it sits in the cytoplasm, and it's bound to HSP, a heat shock protein, and there's a couple other proteins that bind to it and make it stay in the cytoplasm. So then when some chemical comes and binds to the AHR, it will then move into the nucleus. And once it moves into the nucleus, it will form a heterodimer with another compound called ARNT. And what I want you to start to think about as I'm telling you this is that these AHR and ARNT are very similar to clock and BMAL. Okay, they bind together in the same way that clock and BMAL bind. In fact, BMAL is brain muscle aren't-like protein because it's a homolog of this particular protein, okay? So they're very similar. So AHR binds to its ligand, it moves into the nucleus, forms a heterodimer with ART, and then once they form this heterodimer, just like clock and BMAL sit down on the E boxes in clock gene or in clock and drive transcription, AHR and ART binds to xenobiotic response elements, XREs, or dioxin response elements in the promoters of certain genes. And some of the genes that they bind to in the liver particularly, or drive in the liver, are these P450 enzymes. Okay, so you can think of this as a way, this nasty chemical gets into the cell, it activates aryl hydrocarbon receptor, drives transcription of P450 enzymes. The P450 enzymes should then come back and metabolize or break down the ligand. So that's the whole idea, is that the ligand basically is self-destruction, right? So the ligand binds AHR, AHR sets into motion this series of events, and it comes back and breaks down the protein that, or the chemical. Two of the most important P450 enzymes that are target genes for the AHR and ART are CYP1A1 and CYP1B1. I want you to hold on to those because I'm going to talk about those when I talk about how there's an interaction with the clock and AHR. So if we look at the structure of the AHR, the aryl hydrocarbon receptor, as I said before, it's very much like clock and BMAL. So if you look at the structure of the receptor, you see that the AHR has two pass domains, pass A and pass B, which are protein-protein interaction domains. This just shows you the similarity between the structure of AHR and the structure of clock. So they're very, very similar proteins. They're pass domain, it's a pass domain containing protein. They're members of the same family of genes. What's really kind of interesting about the pass domain family is you can divide the pass domain proteins into two groups. They can call them the alpha class and the beta class. Basically, the alpha class you can think of as sensors, environmental sensors, or sensors like HIF1 alpha is a sensor for hypoxia. Clock is a sensor for maybe for NAD and things like that. But these guys are sensors. And then the beta class are binding partners that help direct. Once one of these guys binds with one of those guys over there, the beta class directs them to a specific place on the gene, in specific genes, to drive transcription. But what we know is that ART3 over there is the same as BMAL. So this class, the alpha class over here, can form partners with beta class members, but they can form. It's not a one-to-one relationship. It's not like clock always binds to BMAL. For example, ART can bind to HIF1 alpha or AHR or SIM1. ART2 can bind to all of these guys. And BMAL1, of course, we know that it binds to clock, and it can bind to HIF1 alpha, it can bind to NPAS2. So they form all sorts of different combinations, and the different combinations determine where they go and which genes they turn on. Okay? So what we asked was, because these guys are in the same family of proteins, is can the AHR binds to BMAL? Because remember, AHR normally binds to ART, and BMAL and ART are very, very similar proteins. So we asked that question, do they bind together? Because if they bind together, then they could have a potential for regulating the clock, regulating the circadian clock. And so we did a pull down, and this is what we found. So this is basically, we did a co-IP, an immunoprecipitation. So we took our animals and we treated them either as control, or we treated them with an agonist of the aryl hydrocarbon receptor. And what we see is that, and then we pull down with an antibody against the AHR and probe for BMAL. So what this is going to tell us is the two of them are sticking together. Okay, as we can see that when we pull down with an antibody for AHR, so everything that bound to AHR comes down, and then we run it all in a gel, and we see that it binds to BMAL. So these two can interact with each other, which tells us that it has a potential to be interacting with the clock, interacting with circadian rhythms. And so I'm not going to spend a lot of time on this. So this is just the regular clock that you've seen before. So we've clock and BMAL1. And so what we're going to hypothesize when we do our studies is that AHR can interfere with this clock and BMAL binding. So AHR can pull these two apart and AHR can interrupt this and interrupt the clock. Yeah.
Can I ask questions right
Yes, please do.
So on the previous slide, do you think that it binds in the same spot as clock, or do you think it binds in like more like an allosteric site?
No, they bind through their past domains. So they bind, I don't know if it's exactly the same, but it's through that particular motif that they bind.
Okay.
So it could be slightly different, but it's that structural arrangement that binds it. Okay. All right, let's see. All right, so what I want to spend the rest of the time talking about is this interrelationship. First, we're going to ask the question, does the clock alter aryl hydrocarbon receptor or dioxin signaling? And then we're going to come back and ask the opposite question, does dioxin signaling affect the clock? And we don't know a lot about this area. This is a very new and emerging area of research. And what we do know is that there does seem to be this reciprocal relationship. AHR signaling affects circadian rhythms, and circadian rhythms affect the ability of the AHR to metabolize dioxins. Okay, so the first question we're going to look at is, does the clock interfere, or does the clock direct dioxin signaling or AHR signaling? So I'm going to tell you the answer is yes. And this is sort of the take-home message of the next several slides. AHR signaling is regulated by the clock. So that's big take-home message. So this is a study that we did in my lab, and we did this in mice. And basically what we did was we looked at the expression of aryl hydrocarbon receptor, 2 clock genes, PER1 and BMAL, and CYP1A1. Remember, that's the target gene for AHR. So what we found, we did it in both the SCN and the liver, and you can see there's and so the solid lines are from animals that were in a light-dark cycle, and the dotted lines are for animals kept in DD, and you should probably all know by now that when you put them in DD, that tells you that it's regulated by the endogenous clock, right? Okay, so we can see that both PER1 and BMAL1 show nice circadian rhythms, both in LD and DD, in both the SCN and the liver, like you would expect. And then we did AHR. And what you can see up there at the top is that AHR is regulated by the clock because it shows a circadian rhythm in LD, and then when you put them in DD, they still see a nice circadian rhythm, although it's not as robust. And it's both in the SCN and the liver. And CYP1A1, the target gene for AHR, also shows a circadian rhythm. So this is our first evidence that AHR is regulated by the clock. So AHR is regulated downstream of the clock. And this is a study that was done by a group in Japan, and they didn't do liver, they did lung. And remember that I showed you on that one slide that benzo-A-pyrene was a component of cigarette smoke. And so there's lots of AHR in the lung, and it's used to detox, or to try to detoxify some of these components of cigarette smoke or other things that you would breathe in. Okay, so it makes sense that there would be this in the lung. All right, so they looked at AHR messenger RNA in the lung, and their animals were in LD, and so we see it's the same thing, a circadian rhythm or a diurnal rhythm in AHR in the lung. And then they looked at the target gene, CYP1A1, and they see a nice rhythm of CYP1A1. And one thing I want to point out is notice that the peak in the AHR occurs here around 17, and the peak here occurs a little bit later around 21, which fits with the idea that this gene drives this gene. Okay? So they looked at CYP1A1, the target gene. They looked at CYP1A1 protein, and they see a nice rhythm in the CYP1A1 protein. They looked at CYP1A1 activity. It's an enzyme. And they see that there's a rhythm in CYP1A1 activity. So again, components of the aryl hydrocarbon signaling pathway are regulated by the clock. They also looked, in this same study, they looked at binding of the aryl hydrocarbon receptor to the dioxin response element. So they took dioxin response element and labeled it with something radioactive, and then they put aryl hydrocarbon receptor in there with it. And what they can see is the binding to its target gene is regulated by the clock. So this band is higher than this one. So there's more binding of aryl hydrocarbon receptor at the time of day when the receptor was present more. So we're getting increased messenger RNA and protein for AHR and its target gene, and increased binding of AHR to the gene elements to drive the activity of CYP1A1. then what they did to really confirm that this was regulated by the clock was they took clock mutant mice. And so the clock mutant mice have a disrupted circadian timing system. And so what they did here was they have wild type mice and the clock mutant mice at 2 different times a day. This is in the morning and this is at night. And what we see is in the wild type mouse, and this is Aryl hydrocarbon receptor messenger RNA. This is what they saw before, higher levels of AHR at night, lower during the day. But look what happens in the clock mutant mice. So you take clock away, there's no clock anymore, and AHR levels drop, and there's no rhythm. So the bottom line here is that Aryl hydrocarbon receptor is driven by the circadian clock, and the gene clock is probably very important for its regulation. So then, in one more study that was done by Q et al. And Toxicity in 2010, they did the same thing. In this case, they're looking at per mutant mice. So they take mice that have altered per gene, right? So here's the wild type animals here, and now they're looking at. So we've shown the previous study was able to show that AHR signaling is regulated by the clock. But AHR, what does it do? It binds to these toxins and it stimulates toxin metabolism, okay? So what they did in this study was they asked, if we mess up the clock, does it alter the ability of AHR to respond to a toxin challenge? So they give the animal a challenge to some sort of toxin, and they use TCDD, which is a really, really really toxic compound. As they ask the question, in the per mutant mice, do they have an altered ability to respond to the toxins? So here's the wild type mice, and we see that here's their response, here's the control and response to TCDD during the day and at night. And you can see they have a much better ability to respond to the toxic challenge at night. But then when they take the, when you look at the per mutant mice, and let's just jump over here. Here's the per mutant mice, and this is the wild type. So they have a small ability to respond to the toxin during the day and a much larger ability to respond to the toxin at night. But if you take the per mutant mice, their response to the toxin during the day and during the night are the same. So if you alter the circadian rhythm of the animal, you decrease the production of AHR, which we showed in the last slide, and there's no more circadian rhythm in the ability of the animal to respond to the toxin. So, to just summarize this, here's the bottom line. AHR signaling is regulated by the clock. So the AHR and the downstream components are expressed in a circadian fashion, and this occurs in many tissues. I showed you a couple examples. Clock mutant mice has decreased AHR expression and no circadian rhythm. There's overall decrease in the expression of AHR and no rhythm in AHR. The per mutant mice lose their circadian rhythms of their response to toxins. So overall, the bottom line is, again, my take-home message, AHR signaling is regulated by the clock. Okay, now I'm going to spend the next half hour, the rest of the time, talking about the reverse. So I'm going to talk about how the dioxin signaling pathway can, in reverse, feed back and interfere with the clock, or regulate the clock. So, the question here is, does AHR regulate the circadian clock? And so I'm going to show you 2 different kinds of studies. And all the studies that I'm going to show you are the ones that we've done in my lab. So, and we use two different methods here. Studies, I'm going to show you studies in knockout mice where AHR is knocked out. And I'm going to show you studies using AHR agonists or toxins. So the first question is, in the first set of studies that I'm going to show you, I'm going to ask the question, if we remove AHR, does it matter? Does their circadian rhythm care? Okay, so do the AHR knockout mice have altered circadian rhythms? And have you seen these, the behavioral? Okay, so I don't have to explain these to you. So, this is what we did. We took wild type mice and we took Aryl hydrocarbon receptor knockout mice. And we put them in, at the top, they're in constant darkness. So you see the wild type mouse, which with the normal mouse circadian rhythm, with getting a little bit shorter every day. And then the Arco mice. Does anybody see any difference?
might be just a little bit longer in
Maybe,
the
yeah.
knockouts?
Yeah, there's not a lot of difference. Maybe a little bit, not a difference. So, when we first did this, we were kind of disappointed because we were like, oh, we really thought that knocking out AHR was going to really mess up their circadian timing system. But what we found was that their endogenous clock, the endogenous rhythm, seems to be okay. It doesn't have a lot of problems. However, when we take the same AHR knockout mice and we do a phase shift experiment where we alter the time of the lights on, we've made it come on 8 hours earlier, and then we count the number of days it takes to re-entrain. What we find is in the wild type mice, it takes around 3.5 days on average to re-entrain to the new light-dark cycle. But the AHR knockout mice take about 7 days to re-entrain. So, to a phase advance, and they have they're okay if you go the opposite way. So if you phase delay the lights on, they're okay. So what this tells us is that without the AHR, the animals may have a little bit of a problem responding to changes in the light-dark cycle, or they get a little bit jet-lagged, right? They have jet-lagged for a longer period of time. So if we fly these mice across the Atlantic, they're going to have a harder time readjusting to the new time schedule. That's the main point there. All right, so it looks like they might have an altered response to light. So what's the next experiment that we want to do? Anybody want to take a guess? We're going to give them a shot of light at night and see if they respond normally. Okay. So, in a wild type mouse, if you give them a light pulse in the early part of the night, they're going to get a delay in their circadian rhythm. It's going to look like this. And if you give them a light pulse in the late part of the night, they'll do a phase advance and you'll get something that looks like this. So we did the same experiment in the Arco mice, and this is what we found. In the Arco mice, we gave them here's the wild type with the phase delay, and the Arco mouse actually has a slightly bigger phase delay. So the animals do have an altered response to light. This one might be exactly the opposite of what we might have predicted from our other experiment, because it took them longer to adjust to the new time schedule, but they actually have a slightly bigger response to a phase delay in light, and we're not sure exactly what that means. At this point, all we know really is that they have an altered ability to respond to light. So then, they did show a little bit of a change in their ability to respond to light. So the next experiment we did was ask if we give a toxin to the animal, can it act like a light pulse? So will it activate the light signaling pathway? And so we did that experiment, and we used we used tryptophan itself, which was a control. We used this compound called FICZ. Remember, this is that ligand that has a high affinity for AHR. And we see really no change. So the AHR ligands can't cause a light-like effect. So we asked a different question, and that was, if we give the animal a light pulse and we give the chemical at the same time, does it block or does it change the ability of the animal to respond to light? So if we activate the AHR, does it change the animal's ability to respond to light? So we gave this drug, FICZ, the AHR ligand, and then we gave the animal a light pulse. And we can see that a normal animal has about a little over a 2-hour phase delay in response to light. In presence of FICZ, it has a smaller phase delay, so it does block the phase delay a little bit. So when this is in a normal animal, so this is consistent with that previous data. If we take AHR away, we have AHR knockout mice, they have bigger phase delays in response to light. So AHR levels are down, they have bigger phase delays. And here, when we activate AHR, so we can think of AHR activity as high, they have a lesser response to light. So the bottom line is that the AHR activation can attenuate the light response, and no AHR, the lack of AHR, makes a bigger light response. So, and then we did the same experiment using 2 different AHR agonists, and that's just what this is showing. This is a different AHR agonist, and we see the same effect. And this is a third different AHR agonist, and we see the same effect. And then we did it in the AHR knockout mice, and we don't see the effect and we see the little bit bigger phase delay. So these are all consistent with the idea that when there's no AHR, we get a bigger response to light, and when AHR is activated, we get a smaller response to light. So then we asked the question, because when we gave these agonists, we didn't inject them into the brain, we gave them IP. So AHR activation could be in the liver, it could be anywhere. So we asked whether or not the AHR activation was mediated directly by the suprachiasmatic nucleus. We actually did this in Martha's lab. You've probably seen this picture before. Here's two little brain slices sitting in a brain slice dish, and we recorded their rhythm of their electrical activity. And here, you'll see a normal rhythm of electrical activity in the middle of the day. If it phase delays, the peak in the activity occurs later, and if it phase advances, it occurs earlier. So here's our experiment, and in the dish we use glutamate as a substitute for light, and we can see that glutamate will cause a phase advance when it's given in the late part of the night, and a phase delay when it's given in the early part of the night. So we've used this, and we gave glutamate or glutamate plus the AHR agonist. And so what we see is glutamate causes normal phase advance like we would expect. When we give the AHR agonist, we get no change. And when we put glutamate on with the AHR agonist, we can block the phase advance. So this is just like what we saw in vivo in the animal. So we were able to block the phase advance of the behavior using an AHR agonist. And here in the dish, we were able to also block the phase advance caused by glutamate.
In vivo, though, there was still a slight response, right?
So we gave those, and that's a good question. So we gave those drugs IP.
Right, okay.
So it could be a dose thing, and we don't know. But they are lipid soluble, so they getting across the blood-brain barrier shouldn't be a big issue. But we haven't really done a really good dose response with the drugs,
okay.
but it could be that. Yeah. Okay. So, and you may have seen this slide before because I stole it from Martha, or part of it at least. But here's light and glutamate signaling, and when I was in Martha's lab, we worked out. I worked on part of this, and other people in the lab worked on part of this, and we were able to show this is the signaling pathway for light. But I want to just focus on this right here. So when light hits the SCN, it activates the signaling pathway, and PER1 goes up. Okay, and this PER1 has to go up, or the clock won't reset. So we decided to try to get at the molecular mechanism by which AHR was interfering with the light response. So we asked the question, if we give light or glutamate and the AHR agonist, what happens to PER1? That's what we asked. So normally it should go up. If we block the light response, we would expect that it won't go up, right? So we did that experiment. And here is, we used light plus an AHR agonist called BNF. So here we see our control, here's BNF alone, here's light, and this is the SCN, although the resolution's not real good. But we see it goes up when you give light, this is PER1, and BNF blocks the effect. So the mechanism by which AHR agonists can block the light-induced phase advance or phase delay is by blocking the induction of PER1. So somewhere upstream of the induction of PER1, AHR is acting. This is showing the same thing for a different agonist. So a lot of times in these toxicology studies, they want you to show it with multiple agonists to show that you get the same effect. So here's the same thing, looking at PER1 messenger RNA by qPCR. We see that light makes PER1 go up, and that is attenuated significantly. Here we weren't able to block it completely, but it attenuates it significantly. So when we activate AHR, we block the light response, and we block the induction of PER1. So then we asked, so we know that the activation of the AHR, the Aryl hydrocarbon signaling pathway, blocks the effects of light in the circadian timing system. But what does it do to the normal rhythm? That was the question. Does it only have effects on the light signaling pathway, or does it also affect the normal rhythm? So we looked at that. And so here's animals where we treated animals with TCDD, and then we looked at the rhythm of PER1 in the SCN, and it has no effect. There's no effect on the normal rhythm. It only has an effect during the light response in the SCN. However, now I want to really focus on this interaction between the SCN and the liver. So normally the SCN drives the rhythms in the peripheral systems, and one of the major places is it drives it in the liver. And the liver is really important for AHR because that's where we get the detoxification of certain enzymes. So we asked this question. Here's our previous experiment where we treated the animals with TCDD, the AHR agonist, and we see no change in the SCN. But look what happens in the liver. We give TCDD, here's the normal rhythm of per1 in the liver, and after TCDD, we see a change. So basically, what we think is going on is that activation of AHR has no real effect on the main clock in the SCN, but it obviously changes the clock in the liver. So if a person or an animal is exposed to high levels of dioxin, what may be going on is that the liver clock and the SCN clock are no longer in sync with each other, because the AHR agonists are messing up the liver clock but not affecting the clock in the SCN other than the light response, but their endogenous clock is okay. So what we think may be happening is that when the AHR agonists are there, the clock in the liver is now not synchronized any longer with the clock in the brain. And that could cause all sorts of problems down the road. We don't know what those problems are. I'll give you a clue at the end, but it could be causing problems. So it's causing desynchrony of the systemic timing system. The one in the brain's okay, but the clock in the liver is not okay anymore. So, what's the mechanism by which AHR agonists can alter this clock in the liver? So we looked into that, and we used the cell line. This is a liver cell line, and we have two different kinds of cells, so it actually works like a knockout mouse, because we've got this cell line that expresses lots of AHR, we've got another cell line that doesn't express it at all. And so what this shows is that if we treat these cells, either the C7 cells or the C12 cells, with an AHR agonist, and we measure this is per 1 levels, we can see that the FICZ, the AHR agonist, has a dose-dependent suppression of per 1. But in the cells that have no AHR, when we give FICZ, it has no effect on per 1 levels. And this is the same thing using a Western blot for per 1. So we treat with an AHR agonist, one or the other, and we see that in the cells that have high levels of AHR, it suppresses per 1 protein, there and there. But in the cells that have no AHR, there's no suppression of per 1 protein. So we believe that the mechanism by which, just like what we saw for the light response, the mechanism here for suppressing the clock or messing up the clock in the liver is by inhibiting per 1. We did this another way. We used a luciferase reporter. So we took the per 1 promoter and tagged it to a luciferase reporter, and we did this experiment. So we took our cells and we expressed clock and BMAL in the cells, and when you express clock and BMAL in the cells, we see the per 1 luciferase levels go up because they drive expression of per 1. And then what we did was we added AHR to the mix, and just by adding the AHR, if we add enough of it, you can see that it starts to come down. And it's because this is kind of an artificial system, so probably not all the AHR stays in the cytoplasm. Some of it can leak into the nucleus and activate transcription. But look what happens when we express clock, BMAL, AHR, and then add an AHR agonist. We see that per 1 luciferase levels drop. So activation of AHR is able to suppress the levels of per 1 in liver cells. So then what we did was we asked whether or not this is occurring by binding to the per 1 promoter. So this is a chip assay. Is everyone familiar with the chip assay? Okay, so basically what we're going to do here is we have the per 1 promoter, all right? And we're going to look at if we activate AHR, do we get binding to the per 1 promoter? Normally we should get binding to the per 1 promoter. So this is our control. This is the dioxin response element for CYP1A1, the normal target gene, okay? So here we have AHR, a little binding of AHR to the CYP1A1 promoter. But look what happens when we add BNF, the agonist. We increase the binding of AHR to the target gene promoter, okay? So that's our control. So then this is the E box. This is 3 of the 5 E boxes in the PER1 promoter. So what we asked was, when we activate AHR with BNF, do we see binding to the PER1 promoter? And what we see here is, if we look here, normally we see clock bounce to the E box. And look what happens when we add BNF. When we add BNF, compare these two bands. When we add BNF, we see, compare these two bands. We see less binding of clock to the E box. And we see an increased binding of AHR to the E box. So what this tells us is 2 things. When we activate AHR, it can actually bind to the PER1 promoter. And clock binding to the PER1 promoter is decreased. So what we think is happening is it's coming in and binding to BMAL1 and kicking clock out. So clock can't be there anymore. And so if there's no clock in BMAL, and so it's acting like a repressor. So if clock binds to BMAL and they bind to the PER1 promoter, we get increased PER1. What we think happens is when AHR comes in, it kicks clock out and binds to the PER1 promoter, but it acts like a repressor, so it suppresses, it doesn't drive transcription of PER1, it suppresses it. So that's kind of what we think is going on. And then, as I've shown you before, this is just showing you that if we treat cells with an AHR agonist and pull down with AHR, then we can find BMAL1. So we know that AHR is capable of binding to BMAL1. Okay. So basically what I showed you are a couple of things. Mechanisms of PER1 induction. There are 2 ways that PER1 is regulated by the circadian clock. One is its circadian regulation. So normally we would see that there are E boxes in the PER1 promoter, and clock and BMAL1 can bind to these E boxes and drive the transcription of PER1. Separate from that, when you give an animal a light pulse, then clock and BMAL1 will be pulled in and binds to the PER1 promoter and acutely increase the transcription of PER1. And that's sort of separate from this. But there are 2 ways to regulate PER1 by the clock. Okay? In addition, when you do light, you also have to get Krebs involved. So light, you activate Krebs, it helps clock and BMAL1 to drive the transcription of PER. Okay. What we think is going on is that AHR activation in the liver can actually block the ability of clock and BMAL1 to bind to the E boxes here. And so it can suppress PER1 levels. And we saw that in the liver. We saw that with the circadian rhythm, because the rhythm, remember, the rhythm was delayed. We saw that delay, and that's probably because AHR is acting as a repressor and not letting PER1 be made. And then down here, when the light response occurs, in the SCN, it may be preventing clock and BMAL1 from binding to the E box and preventing the light induction of PER1, which we saw. Okay. So we think what's really going on, as I alluded to before, was the AHR, because it's so highly expressed in the liver, when it's activated in the liver, it alters the circadian rhythm in the liver, and what it leads to is a desynchrony between the clock in the brain and the clock in the liver, and we believe that this can lead to pathology, can lead to disease. So what kind of disease might we be talking about? Well, there are a couple of, we know from epidemiological studies that I've showed you that AHR activation or exposure to dioxins can cause carcinogenesis, it can cause all sorts of things I showed you in the beginning, and one of them was diabetes. And so what we've been working on lately in my lab is asking the question, does the AHR alter the circadian rhythm of glucose metabolism? So I'm going to show you just a little bit of data from my lab. This is wild type mice and the Aryl hydrocarbon receptor knockout mice. And this is blood glucose levels. And so blood glucose levels measured at 4 different times of day. And we can see in the wild type mice, we see a nice circadian rhythm in serum blood glucose. And then look at the aryl hydrocarbon receptor knockout mice. So they actually have better blood glucose levels, but their circadian rhythm is suppressed. Okay? So if we look then at this gene, peroxisome proliferator PPR alpha, if we look at that in the wild type animals, we see a nice circadian rhythm in PPAR alpha. If we look at the aryl hydrocarbon receptor knockout animals, we see that's flat. It's not significant. So basically what this shows is that the AHR knockout mice have altered circadian rhythms in serum glucose and in PPAR alpha, which is a gene that's important for glucose metabolism. Okay? So that's about as far as we've gotten. So what we are doing now is we're looking at, we have done one more study that I didn't put here. If we put animals on a high-fat diet, they get diabetes. Okay, they get high serum blood glucose and diabetes. Okay, so when and we now know that the aryl hydrocarbon receptor is involved, and that's about as far as we've gotten. So to go any farther than that, I'd probably be making things up. But all I want to say here is that AHR, when you knock out AHR, you alter the circadian rhythm in glucose metabolism, and you alter the expression of genes that are important for glucose metabolism. And we believe that this may be an important mechanism that is a part of how dioxins can cause diabetes. So that's where we'll just leave it there. So just to summarize the last part, the aryl hydrocarbon receptor knockout mice have normal baseline circadian rhythms, but they do have an altered light response. When we activate the AHR using toxins or agonists for AHR, we can attenuate the light effects in the SCN. The activated AHR inhibits PER1. AHR can do, and how does that happen? Well, AHR can bind to BMAL1, and it can replace clock on the E box. So it displaces clock. So clock and BMAL can't bind to the receptor and can't drive the expression of PER1. And then finally, the aryl hydrocarbon receptor knockout alters the circadian rhythms in liver metabolism, especially glucose metabolism and serum blood glucose, which may provide the link between circadian rhythms, the aryl hydrocarbon receptor, and the development of diabetes. Because we know both circadian rhythms and aryl hydrocarbon receptor or toxin exposure can cause diabetes. Altered circadian rhythms can cause diabetes, and toxin exposure can cause diabetes. And we believe that the aryl hydrocarbon receptor and its function is going to be central to linking the circadian rhythms to diabetes. Okay? So are there questions? That's all I have. Oh, that was the slide. I'll just go through this on the last slide. So what we showed was that we get a big effect of when you expose the animal to that dioxins. We get a big effect on the clock in the liver and a small effect on the clock in the brain, which may then, we believe, lead to, it's not doing it, a disruption. So what, we get an effect on the clock in the liver, very small effect on the clock in the brain. As I said before, we think that this disrupts the synchrony between the clock in the liver and the clock in the brain. And we think that once you've disrupted this synchrony, it leads to pathology. And in the case of liver metabolism, the pathology that could be involved is the altered glucose metabolism that leads to diabetes. So altered glucose metabolism, we get metabolic syndrome and diabetes. So that's all I have. So I'm happy to take any questions, discussion, whatever.